Hello, my name is Grant Hartung, and welcome to my talk entitled Capillary Density Induces Microvascular Biases in Bold, Insights from Realistic Vascular Modeling. Large draining veins are well known to impart artifacts in bold fMRI, as previously demonstrated here in the visual cortex where the pattern of neural activity is the same across cortical depths, yet the bold response in blue has artifacts when measured near the pial surface from large pial veins. While these biases related to the macrovascular anatomy are well known, with increasing fMRI resolution, it is also important to understand possible microvascular biases caused by regional variations in capillary density. So here we ask, how does the microvascular anatomy shape the bold response within the cortex, specifically investigating how variations in capillary CBV across cortical depths may impact how the bold response amplitude varies across cortical depth. The microvascular architecture varies across cortical depth as clearly shown here in S1, but not in neighboring M1 in the classic study by Duvernoy using human vascular staining. Here, the middle layers have an increased capillary density, which is also seen in V1, but not seen in neighboring V2. Indeed, many primary sensory areas are granular cortex, and it has been shown in mouse data that the fractional vascular volume, or CVV, has a peak in the middle of the cortex. Given that a high capillary density is often found near the middle cortical layers, here we ask, could this microvascular feature cause the bold bump, or the local peak of the bold response amplitude in the middle cortical layers, as seen in many studies, perhaps most clearly observed in humans by Koopmans et al. In 2010? Unfortunately, answering this question by identifying microvascular biases is challenging. Isolating the vascular architecture from the underlying neuronal architecture is tricky because we know that higher capillary density is expected in regions with higher neuronal density and higher metabolic needs. This is shown nicely here by Blinder, who quantified and compared neuronal and capillary densities, and both exhibited a peak in the middle cortical layers. This means that the peak bold response in the middle depths could indicate more neuronal activity. Moreover, bold responses are coupled across cortical depths through the vascular hierarchy, as shown here by Havlicek in 2020. Even under the assumption that microvascular responses to neural activity are localized, the veins integrate signals and spread the bold response across cortical depths. Fortunately, simulations can be used to elucidate how vascular architecture influences the bold response, but with the important feature that more realistic vasculature leads to more realistic coupling between the vascular architecture and the bold signals. So here we use a simulation approach based on vascular anatomical network, or VAN modeling, that represents all blood vessels within a single voxel here from a mouse brain. These arteries, capillaries, and veins represent the vascular anatomy through an interconnected network of blood vessels. For the dynamics, we use as inputs active arterial dilations taken from 2-photon recordings of single vessel dilations responding to sensory stimulation, representing neurovascular coupling, from which we can simulate the resulting changes in blood flow, pressure, and volume dynamics. Once the flow and volume are known, we can compute the blood oxygenation dynamics in the vasculature and surrounding tissue using our expanded 1D 3D coupling model. We then calculate blood susceptibility and compute the magnetic field offsets around the vessels to simulate a dynamic bold response. It is important to note that in order to simulate the dynamic bold response with larger vans that span the entire cortical thickness, we had to overcome the computational burden from previous methods based on unstructured tetrahedrons that contour to the blood vessel walls. This required the adaptation of a steady-state 1D 3D coupling that does not require contour meshing, and enables the use of structured hexahedrons that are similar to voxels as mesh elements. This generates accurate computations on meshes even with large hexahedral dimensions, which is especially important when 97% of the simulation domain is the extravascular mesh, so reducing the computational burden for this part of the simulation is very important for our large dynamic van simulations. One may ask, however, why was it necessary to expand the 1D 3D coupling method to dynamic simulations, which take substantially more effort than baseline or steady-state computations? Our expansion of these simulations to dynamics is not just a benefit or sanity check, but rather is a fundamental part of our investigation to observe transient effects. These differences come in the form of different peak timings and different amplitudes, both of which are shown here to vary across different depths. These different transient effects observed through timing and amplitude characteristics of the response may hold valuable information on the function of different regions, but these assumptions are predicated on the hypothesis that stronger or earlier signals in a layer indicate the primary response layer for a region. It is important to note that these layer-specific timings in bold signal are not an input to our model, but rather an output, based on the vascular architecture as discussed next. Here are the results. We first demonstrated using two vans reconstructed from mouse somatosensory cortex that mid-depth peaks are observed in the capillary density or baseline CBV, here termed as CBV0, and also in the bold response. This peak in the bold response in the middle cortical layers is not simply reflecting a larger change in arterial dilations recorded in the two-photon microscopy data used as input to our model. To verify this, we performed the same simulation using the exact same arterial dilation trace and amplitude and timing across all depths and observed the same mid-layer bold peak. This means that the observed bold peak in the middle layers is a function of vascular architecture and not input stimulation design or varying neuronal stimulation across layers as mentioned on the previous slide. But the question still remains, although we see a bold bump in the depths alongside a peak in the capillary density, how do we know whether the local peak in capillary density is the cause of this bold bump? To test this, we sought to move the location of the peak capillary density and see whether the bold bump moves along with it. To do this, we developed a method to synthesize Van models with a specific vascular architecture. We previously applied Van synthesis to build a human Van model. While it is possible to reconstruct the full microvascular tree in vivo in small animals using invasive microscopy, it is more challenging to reconstruct the full 3D vascular network in humans. So instead, we can synthesize Van models based on vascular distributions taken from the literature. And here's an example of a realistic human Van synthesized from the published Duvernoy data. A similar approach has been adopted by Natalia Petridu's group. Van synthesis is also helpful because it allows us to parametrically vary individual properties of the vascular architecture to evaluate the impact on the bold response. As we see here, we can feed into our synthesis algorithm two different histograms of capillary density across cortical depth to generate synthetic Vans with all properties held constant with the exact same arteries and veins, except that each has a dense capillary bed at a different cortical depth. Here's a brief summary of our vascular synthesis algorithm, which we have previously published. Our method is built on constrained constructive optimization that builds a vascular network stochastically by adding segments with the pre-assigned geometrical properties until the desired CBV is reached. The final result, after all synthesis stages are complete, is a fully connected Van ready for dynamical simulations that achieves the targeted statistical properties. As a quick validation, because our synthetic Vans have never been used in similar bold simulations, we generated 5 Vans from the statistics of one of the reconstructed Vans. We simulated the bold on the 5 synthetic Vans and observed a similar time course and amplitude as the 2 reconstructed Vans, and showed that synthetic and real Vans yield similar bold responses, which is reassuring. Next, to investigate microvascular biases, we created 3 unique capillary profiles with higher capillary density in the lower half, middle third, and layer 4 for different models, respectively. Each of these 3 models has a fixed backbone of arteries and arterioles, veins and venules, overall capillary baseline CBV, and an overall number of vessels. What we varied was the capillary locations to fit the desired profile. Here we see that in each model, the baseline capillary CBV profile in colored lines has a distinct peak in the desired region. The bold profile is then computed using all vessels, which shows 2 peaking regions. The first peak is near the pial surface from the large pial veins, and the second is the peak in the high capillary region, with high baseline capillary CBV 0. We also calculated the statistical correlation between the CBV and bold in each group and found that the change in bold and capillary baseline CBV have high correlation within each model, but relatively low correlation to other models. There is, however, some overlap between the lower half bold and layer 4 CBV 0, which we attribute to the considerable overlap between the baseline capillary CBV profiles for these 2 models. Our model does have limitations, however. For instance, we have an exaggerated capillary density profile, much larger than likely would be found in vivo. This was, however, intended to highlight the capillary impact on the bold profile more than the noise in the capillary CBV profile naturally, which is an inherent part of the synthesis algorithm. We also noticed a peak bold response in the mid-depths, even in our control model using a uniform capillary density. We attribute this, however, to a gradient in baseline oxygenation across depth. As a sanity check, we verified this gradient exists across multiple types of boundary conditions aimed at eliminating tissue boundary effects, so this gradient is likely an effect of the vascular architecture that should persist in vivo, as previously suggested. We also noticed that this same peak in the lower layers is absent in a corresponding upper half profile. Moreover, the uniform capillary baseline CBV does not correlate well to any of the other bold profiles. Altogether, this suggests the microvascular structure imparts a bias impacting the bold profile, but the exact nature and realism of this effect needs to be further investigated. In conclusion, microvascular architecture imparts a bias on the bold signal. Specifically, the change in bold tracks the baseline capillary CBV closely, as expected. Van synthesis can then investigate the impact of vascular anatomy on the bold signal. This enables creating bands that are unable to be reconstructed and allow isolating independent vascular properties. Capillary baseline CBV may also explain some findings in later fMRI. For instance, capillary baseline CBV may impart stimulus-independent bold bias, but this baseline CBV may also be linked to neuronal or metabolic demand, so this bias may also reflect underlying neuronal structure independent of neural activity strength. I would like to thank the following people and our funding sources for their support.